Hi, welcome to today's class. For today's learning purposes, we'll be talking about direct KOH mounts. I want to thank our students and our viewers for supporting our YouTube channel by subscribing, our returning subscribers. Thank you so much. And for those of us that have not subscribed, what are you waiting for? Just click on the button right there and subscribe so you'll be the first to be notified on more educative content such as this. So for today's class, we're talking about direct KOH mount. Direct KOH mount. Now, KOH means potassium hydroxide. Now, it's a solution just as we have here. This is KOH. It's used to do microscopy for fungi studies. So today we're talking about how to carry out KOH mount. How do we use KOH to identify fungi present in samples? So for today's learning purpose, I'll be demonstrating to you how to carry out KOH mount. But first of all, we need to know what are you expecting to see, the shape of the fungi and how it looks like. The use of using KOH, which is potassium hydroxide, you must remember that it is corrosive, because when you are working with it, you have to know the danger involved in different reagents you use. Now, fungi are a group of or a class of microorganisms that grow at room temperature. Now, we have different kind of samples that we can use KOH. You can use a hair sample, skin scraping, nail scrapings, name it. So when carrying out KOH, you obtain the sample. Like for today's learning purpose, we have hair sample. Now when you carry out, you have to obtain the sample into a sterile paper, just as we have here. Now the shape of these fungi, like when you view KOH preparation that has fungi under the microscope, you may see something like this. Now, this is a hyphae. This is a hyphae. This is aseptate hyphae. It doesn't have septa, so it's aseptate. Now, if it looks like this, this is septate. Septate hyphae. Now, this, they are the septors. They are the septors. Now, if these septors thicken, these demarcations now, if they thicken and separate away from the hyphae, they form what they call Arthoconidia. Sometimes you see something like this. When they thicken and separate away from the hyphae, they form this. Now is called Arthoconidia. They are called Arthoconidia. So you can actually see Arthoconidia in your hair sample. You can see septate hyphae, you can see aseptate hyphae. Now, how do we carry out KOH mount? The first thing to get a clean grease-free slide. You get a clean grease-free slide, you put a drop of the KOH solution onto the clean grease-free slide, and then you obtain the hair sample, for instance, the hair sample as we have today, and then you cover with the cover slip. Now, the hair sample contains keratin. For you to be able to view this hair sample under the microscope using KOH, the KOH will have to dissolve the keratinous layer or the keratin present in the hair sample. So you have to allow for a while for the keratin layer to clear off. Usually for hair sample, it takes between 5 to 10 minutes for the keratin layer to clear off. What you do, you place it into a sterile petri dish, moistened with wet cotton wool, and then you allow for a while. But if you can't do that, you can fasten the dissolving of the keratinous layer by the use of heat. So you can use heat to dissolve the keratinous layer, and then you can move on to viewing the mount, the KOH mount. So that's what we're going to do today. We're going to use heat to actually fast track the dissolving of the keratinous layer of the hair sample, so to enable us to see the fungi or the hyphae or Arthoconidia if present. All right. So to carry out a KOH mount, you get your clean grease-free slide, and using your pipette, you get a drop of the KOH onto, you can put, I usually put 2 to 3 drops of my KOH onto the clean grease-free slide. Can put 2 to 3 drops using this small pipette. And then using the applicator stick, use the hair sample. I'll get some quantity of the hair sample onto the KOH, which I have placed on my slide. Okay. So you see, I've placed the sample, hair sample, onto the KOH mount. You know, to avoid drying up while heating, I usually add more drops to avoid my KOH mount drying up. So you may want to add some more drops to avoid the preparation drying up while dissolving the keratinous layer on the hair sample. So I'll cover now my cover slip. So now I've covered my cover slip. The next thing is to heat to remove the keratinous layer of this hair sample. But note, do not allow the KOH to boil. Remember I said that KOH is corrosive. You just heat for a few minutes and you remove. You heat for a few minutes, you remove. Don't keep it for a long time. Don't leave it on the fire for a long time. So see, I just pass over flame intermittently. Don't allow it to boil. It can actually burn your hand. So you don't allow it to boil. It can get so hot and burn you. So now, you see that the keratinous layer of the hair sample is out. So this is how it should look like. This is how it should look like if the keratinous layer is all dissolved. You can see. Huh? See how clear it's looking. So we have finally cleared the keratinous layer. Now it's time for us to view using our ×40 objective lens to check for the presence of fungi in this sample. So we have now focused our Carage mount, our direct Carage mount, and as you can see under the microscope, these are clusters of Arthoconidia. So as you can see, they are just everywhere. So like I told you earlier, Arthoconidia are detached hyphae that are thickened and then separated from septate hyphae. So these shiny things you're seeing now, that look like pores now, they are arthropods now. They are thickened septate hyphae and are separated away from the whole bunch. So now, as you can see, we have clusters of them under the microscope. If you look very well, you see them, clusters of Arthoconidia. See a lot of them. So this is how they look like under the microscope. Now, if you've watched this video to this point, I want to ask you, why not subscribe? And why not subscribe to our YouTube channel and share this educative content to your fellow students and anybody whom it may be of help to. If you have any question about today's class, you can just comment, ask a question on the comment section, and I'll be there to answer your question. Thank you for today's class.